Today, I'm going to talk about agent-free pigment and flow path imaging as a new and fast retinal imaging modality. And I'm Yasuno from Computational Optical Group at the University of Tsukuba, and this study is in collaboration with Topcon and Tokyo Medical University. The subject I'm going to talk in this presentation is the fifth and fast imaging modality. What is the fifth and fast imaging modality? To think about it, I would like to start from color fundus photo at the first and fast imaging. And then the second might be FA and ICGA, and the third can be FAF, and then recently we have the fourth, OCTA. And what is the fifth? It might be a comprehensive non-invasive angiography. And this non-invasive angiography should be sensitive to circulation, vasculature, tissue abnormality, and RB abnormality. So, how we can achieve this requirement? To achieve this requirement, we are interested in three key technologies. The first is the Jones matrix polarization sensitive OCT, and the second is pigment and flow imaging, and the third is Lissajous scan OCT for motion-free imaging. I'm going to explain them one by one. The first key technology is the Jones matrix OCT. Jones matrix OCT is an extension of OCT which can measure the three-dimensional distribution of the Jones matrix in a sample. Jones matrix is a purely physical and mathematical quantity, so it might be out of your interest. But here I would like to emphasize that the Jones matrix possesses all of the optical properties of the tissue. So once we measure the Jones matrix, we can derive a lot of clinically useful images. It can include a high-sensitive OCT, local scattering image, OCTA, and flow contrast. In addition, by applying the polarization analysis, we can have the birefringence contrast, which is known to be sensitive to collagen, and also polarization uniformity image. It is known to be sensitive to melanin. In this particular project, what we are interested in is flow contrast, OCTA, and also melanin contrast through the polarization uniformity. Which kind of information we can obtain from the clinical cases by using the Jones matrix OCT? Here it's an example of the case of PCB. Here in this image, the OCT is appeared as gray, and the melanin, that was the information extracted from the polarization, is appeared with green. And at the top of pigment epithelial detachment, we see a gold particle, which is flow. So, for example, here in this case, we see a huge cluster of abnormal blood vessels at the top of pigment epithelial detachment. Now, even on top, the melanin has gone from the RPE. So far, we are quite happy about the result. But as we are doing the clinical study by using the Jones matrix OCT, we gradually find that the information we can obtain from the Jones matrix is a little bit too much. Then we started to think of properly reducing the information of the Jones matrix OCT. And this part of the project, we called it as pigment and flow imaging, path imaging. Pigment and flow image is an en-face image which is simultaneously sensitive to the abnormalities of blood vessel and also retinal pigment epithelium. To create the pigment and flow image, we are going to use two types of OCT volumes. One is OCTA and the other is the polarization uniformity. OCTA is used to determine the pixel brightness. From the polarization uniformity, we compute the cumulative amount of melanin above the blood vessel, and then this is used to determine the pixel color. By fusing these two volumes, we are going to have this pseudo-color OCT volume, and we are going to watch it from the top. Finally, we have this en-face path image. In this path image, the retinal vessels are appeared in hot color like whitish yellow because there is no melanin on top. But if there is a melanin on top, the blood vessel is appeared in blue. Let me show a path image of an AMD case. In this case, we can see a clear abnormal blood vessel at the center of the retina. We surely can say that it is abnormal because of its shape, but at the same time, its color appearance is also abnormal. It appeared with orange color, means on top of this CNV, the pigment has already been gone. In addition, by zooming it up, we see a small blue appearance here. This means there is a residual of retinal pigment epithelium at this point. We did 3 times of ranibizumab injection to this subject and took the path image again. In this path image, we can confirm the perfect recovery of the retinal pigment epithelium. So far, we are quite happy about the path image, but at the same time, we have found several issues. For example, we see a lot of horizontal line artifacts. By zooming this part up, even we can see several discontinuities of the blood vessels. To make the path image as a real and fast imaging modality, this issue should be overcome. How we can solve this issue? Our solution is Lissajous scan OCT. Lissajous scan is a new scanning protocol of OCT, and this scan starts in the en-face plane as a line, and this line becomes ellipse, circle, and ellipse again, and line, and ellipse, circle, ellipse, and become the line again. And we are going to repeat this process 4 times to obtain a single volumetric OCT data. Why we use Lissajous scan? Because it has a very good property to correct the eye motion after the acquisition. Any small portion of the Lissajous scan might have the 4 overlapping regions with the other small portion of the Lissajous scan. And these 4 overlapping regions can be utilized as landmark to co-register these 2 portions. Since small portions of the Lissajous scan can be regarded as motion-free, the finally registered entire volume of the Lissajous scan also can be regarded as motion-free. By combining Lissajous scan and OCTA, we can visualize fine blood vessels. In this particular case, the raster scan didn't work. There are a lot of motions with this subject, and there is no significant image obtained. But even in this kind of bad case, Lissajous scan provided us a more than satisfactory image quality. So finally, we can combine these 3 technologies: Jones matrix OCT, path image, and also Lissajous scan. By combining it, we are going to have the 5th and fast imaging modality. To demonstrate this 5th and fast imaging modality, we measured a case of myopic CNV. In the OCT and fast image, we see hyperscattering appearance, but we didn't see anything in the OCTA. However, in a path image, we see an abnormal color of the blood vessels at the location of hyperscattering. Another interesting appearance is this polyp in a path image. So here it's very evident in a path image, but we cannot see it in an OCT, and also we cannot see it in an OCTA. By checking the cross-section of the OCT, we see a very moderate abnormality at the RB complex, and also we can see hyperpenetration. So this might be the indicator of a very small abnormality of the retinal pigment epithelium. But this is not that much evident in an OCT, but it is quite evident in a path image. Let me conclude my talk. En-face imaging is advancing. It starts from color fundus photo, goes to FA and ICGA, FAF, OCTA, and now we have the 5th en-face imaging modality, multi-contrast en-face OCT. The hardware device for this multi-contrast en-face OCT is available, so we hopefully can report more interesting clinical results in the future conferences. Thank you.